To understand protein structure and function, it's necessary to actually isolate, to purify proteins, and then to characterize them. Because proteins vary in their function, their size, and their structure. In order to understand a particular protein and how it does what it does, you have to purify it and you have to characterize it in detail using biochemical methods. That leads to the ability to both manipulate the structure and function, because we can now do a lot of variation that involves changes in DNA sequence that changes protein sequence. And these integrate to provide insights and also to generate new applications for using proteins. So what are the sources? Well, first of all, you have to have enough of a protein in the original source in order to purify and characterize it. And what that meant was that things like eggs or milk or blood were where the first proteins came from, the first ones that we knew anything about. And also the ones that were purified, there were large amounts and they were easily purified. So examples are hemoglobin in blood, egg white in lysozyme, casein in milk, and a wide variety of other proteins, and they had very diverse functions. And so there was this sense of proteins as very different, for example, than DNA. But that will be for another course. So proteins are diverse. They have 20 components. They have different sizes, different shapes, and they carry out all these different functions. And these first proteins were all well-folded, what we call globular structures. They're folded into a nice compact form. On the other hand, intrinsically disordered proteins are often lower in amounts, and they are much harder to purify. Many of them have not been purified as entire proteins. Pieces of them have been purified. And as a consequence of this, globular proteins have strongly affected our view of protein structure as a whole. We think of protein and we think of these nice, pretty crystallographic structures and well-behaved proteins, when in fact, probably the majority of proteins do not fall into that category. So we have basically used organisms and the proteins they contain for literally millennia, for generations and generations. Fermentation using particularly yeast has gone on for years because there are specific enzymatic activities in yeast that either release CO2, as in the rising of bread, or lactobacillus that when you mix with yogurt they create a metabolized form of milk that is very edible and it lasts a little longer than uncultured milk. With wine, beer, all of these have used fermentation of organisms that have specific enzymes in them that produce this fermentation process. And we were sort of dependent on all these organisms to give us proteins of interest. And what changed was the discovery of DNA and then the ability to take DNA and isolate pieces of it that allowed us to cut out coding regions of DNA. Again, this is a topic for another course. And introduce these coding regions for proteins into simple organisms. And that allows the generation of very large quantities of specific selected proteins. You pick the one that you want, you take its coding region, you put it into a plasmid, you grow it in a bacteria, you express it. And even better than that, some proteins are really hard to purify, and we'll talk about how you do that later. But if you add special sequences that have the property of binding to something, a histidine tag as an example, often abbreviated as a his tag, that histidine tag binds to nickel. And so if you have something that allows you to bind that tag, then it helps in the purification of a protein. So there were two more advances that were very important in purifying a wide range of proteins. One of those was the ability to grow cells in culture, and specifically to grow bacteria in hundreds of liters in fermenters, and there's one that's illustrated here. And they range in size. Fermenters can be a liter or they can be 1,000 liters. This one happens to be 100 liters. And mammalian cells are generally not amenable to growth in such large volumes. Generally, they are grown in much smaller volumes. But in both cases, you grow the organism in media, and the cells that are being grown are designed for the overproduction of the protein that you want. So this combination of the ability to introduce specific sequences that encode a protein in very large amounts using plasmids and used as carriers of DNA sequences. I just want you to have in your mind that there are ways to enhance the amount of protein produced. And these ability to grow in large quantities and to handle proteins and DNA in new ways has allowed protein expression in both prokaryotic and eukaryotic cells of proteins that historically, even 10 years ago, would have been very difficult to isolate. So with eukaryotic cells, you can either isolate the cells from the tissue in which it's found, whether that's liver or skin or whatever. And historically, a lot of things were isolated from bovine sources, from cows. And that was because you could go to the slaughterhouse and you could get liver, or you could get spleen, or you could get muscle. And so many proteins were first isolated from animals where you could get large quantities of material. And it turns out that you can also culture tissues. That's much, much more difficult than just taking a single cell organism and growing it, because you have to adjust oxygen and pH. There are complex growth factors that are needed, but it does provide the ability to do more complex experiments and to isolate a greater variety of proteins. And sometimes you get overexpression, sometimes you get less expression when you put them into cells, but it's still an opportunity to get more of the protein that you want. And it's possible also with eukaryotic cells to do some analysis in vivo. We'll talk later about cell sorting methods where you can measure the specific properties of the cell and using fluorescence microscopy and antibodies to highlight specific components, and we're going to talk some more about that later. So if you were planning to purify a eukaryotic protein, what information would be helpful in designing exactly how you were going to go about it?